Good evening, my dear students.
Good evening, sir.
Good evening, ma'am. Good evening, Ajay.
Good evening, sir.
Are you all right now?
Sir, no, sir. Still not well, sir.
Shall we start, Ajay?
Yes, sir.
Okay. Okay. Last class, what we have discussed, in the part 3, some mutation and what are the type of mutations and what are the changes which occurs within the chromosome triads. That things already we discussed. Today we are going to discuss about the continuity of the molecular base of the inheritance. So that what is that? See that chemically induced mutations. Previous one spontaneous and induced mutations also already we discussed. In this part, chemical induced mutations. It comes under the getting induced mutations. See, the first discovery of a chemical mutation was made by Carlet Orbach and the mutagenic effect of mustard gas. Okay, so this is the first one which was used as a chemical weapon in World War I. Mustard gas was first. They were used for the chemical induced mutation purpose. Why they are? It is nothing but the bio war like that. Okay, here the chemical weapon used in the First World War. Okay, so what are the chemicals that were used in the First World War and what are the chemical substances they can induce the mutation? That things we are going to discuss here. So next one is nitrous oxide. Nitrous acid. It's a deaminizing agent which changes cytosine to uracil, guanine to xanthine, and adenine to hypoxanthine. These are the changes. What is it? These are the cytosine to uracil. So deaminating means what is the meaning of deaminate? Removal of amino group from the molecular structures. As a result, what happened? No, one molecule change into another one. Okay, such type was induced by the one of the chemical is called as what is the nitrous acid. Okay, so it is deaminating agents which changes cytosine to uracil and guanine to xanthine and adenine to hypoxanthine. So these are the important things. Okay, these are all our chemical induced mutation, particularly nitrous acid. Okay, so go to the next one, and this is the changes. See that cytosine. Okay, this is the molecules. Deaminase means already in our this is the position. Okay, amino group is here. So this amino group is completely removed. See the next structures. You can see it here. Only the oxygen is occupied. So as a result, what happened? So cytosine is converted into uracil by the process called as deaminations. Okay, so this is the important things. Next one is, say that same thing, cytosine in uracil, and another one is called as what is that? 5-methyl cytosine to thymine. So these are all comes under the headings known as chemical induced mutations. When the chemicals or nitrous oxides activate upon the cytosines, what happened? It may modified into uracil. This is what happened already in oral boards. NH2 is replaced by amino acid deamination process. And also the oxygen is occupied in the particular carbon position. Okay, so go to the next one. Alkylating agents. Another one is also called as alkylating agent. These are chemicals that donate alkyl groups such as methyl or ethyl. Methyl means CH3, ethyl means CH3, CH2 group to nucleotide bases. Okay, examples included ethyl methyl dilphonate EMS, nitrogen mustards, diethyl sulfate DES, and dimethyl nitrosamine DMN, etc. These are the alkylating agents. Alkaline means donate the alkylate. That is very, very important in this part. Base analogs. One class of chemical mutagens consists of base analogs, chemicals, which is structure similar to that of any of the 4 standard base of the DNA molecule. Okay, the structure will be base analogs means, what does it mean? one structure, one class of chemicals is also similar to that of the same component of the DNA. What is the nitrogen? There is, our 4 adenine, guanine, cytosine, and thymine is also. Okay, that is things. DNA polymerase cannot distinguish these analogs from the standard bases. DNA polymerases is also one of the enzymes, isn't it? It's also comes under the things bases analogs, but we cannot able to distinguish this component. Okay, so these are the 3 things. Mutagens. So chemical mutation is the, and particularly nitrous oxide. So nitrous oxide, what happened? There is a changes. And then second one is called as, what is it? Alkaline agent, alkaline donation, base analogs. These are the 3 headings already I discussed. So if the base analogs are present during the replication, they may be incorporated into the newly synthesized DNA molecules. Suppose these type of base analogs are present in it, what happened? They may be incorporated with the newly formed DNA molecules. For example, 5-bromo-uracil. So uracil is the component. The 5-bromo means bromine components addition with the 5th carbon atom. See that is an analogs of the thymine. It has the same structure as that of thymine, except that it is as a bromine. So uracil is also similar to that of the thymine. But what happening here? So we are introducing the 5th 5-bromo, 5th carbon atom occupied bromo. So that's why they have given us a Br here, a symbol letter bromine. Atom on the 5th carbon atom instead of the methyl group. Already we studied that things also, isn't it? What is the meaning? Methyl is present in the thymine. Okay, that group is replaced by the one of the analogs is called as bromines. That's why they called, hence they called 5-bromo-uracil, because 5th carbon atom occupy which one? Methyl group. Instead of methyl group, bromine is occupied. Normally 5-bromo-uracil pair with the adenine, just as a thymine does, but it occasionally mispair with the guanine, leading to the transition process. Actually, the uracil always, what is that? Adenine always combined with the molecule of thymine. But in this category, when the bromo-uracil is formation takes place, the uracil combined with the molecules of thymine, but it does not happen. Instead of thymine, guanine, leading to the transition process. As a result, the changes occurs, mutation occurs. That is called as chemical mutation. Okay, so this is very, very important. Another mutagenic chemical is 2-aminopurine, 2-amino. So 2nd carbon atom, you must note it down. See that, which is a base analogs of adenine. Normally 2-aminopurine base pair with the thymine, but it may mispair with the cytosine. Normally purine base pair with the thymine, but it may mispair with the cytosine, causing a transition mutation. Transition, see here, A is always joined with the thymine. It's a correct order. Okay, so mispair, how it will be? So adenine combined with the molecules of cytosine. This is also a, what is a mispair actually? Okay, so this is it happened when you are adding the chemical is 2-aminopurine. When you are adding the aminopurine, the normal bond, it is not possible. Instead of this one, we will get it this type of mispair. Okay, so don't forget, my dear. And then acridine dyes also one of the chemical substances that were used for the chemical mutations. These are tar-derived heteroaromatic flat molecules. Flat molecules means what is that? The chain structure will be a straight in manner. There is no any up and down movement. The flat molecules. Acridine example: acriflavine, proflavine, euflavine, acridine, orange, and ethidium bromide and dioxin. These are all considered the gettings known as acridines. And also it has a specific dye because orange, acridine orange. enter the DNA chain in between two base pair and cause deletion or additions. Already we discussed yesterday class, we saw it. Addition means a particular nucleotide will be removed. That is called as deletion. Addition means if you are adding any one of the genetic code or nucleotide in between, anywhere. Okay. So of a few nucleotide causing frame shift or Gipresic mutations, this is called as, it's another technical term or a scientific term has given here. So frame shift means a particular codon completely removed. Frame means 5 to 10 codons completely removed and alternative with the another one. Okay, that is called as frame shift completely. Okay, see that one, thymine. When you're adding 5-bromouracil, see that the fifth place that is occupied by the 5-bromouracils. This is also a keto form. This is the enol form. See here, OH is the, so this is called, comes under the enol, alcohol, OH. Here only the O only present, that's why it is concentric is known as C double bond O. So that is keto group. So see here, 5-bromo keto forms adenine and 5-bromo enol form guanine. So this is the difference between the chemical substance. So see the bond also here, double bond is the, here triple bond is also there. Okay, so this is very, very important. Hydroxylamine. So another one is called as hydroxylamine. The hydroxylamine add a hydroxyl group to cytosine. Okay, converting into hydroxyl aminocytosines. Hydroxylamine plus cytosine, hydroxylamine, cytosine. Additions. It is also changed in the nucleotide sequences in the particular DNA molecules. Okay, see that thymine, same thing is also given here. Okay, and the second point is, second main component is radiation. Already you know very little about radiation, is nothing but the rays. Okay, comes from any part of the body. Major source is, ultimate source is, what is that, you know? Sun is the ultimate source of energy. Already we studied so many years. Here the radiation is the, see that there are so many types is the ionizing radiations, non-ionization. What is my ionization mean? Same as ions from the particular area or particular place, particular component. The ionizing radiation such as gamma rays and X-rays, depending on the energy of the radiations, can create free radical that result in problem ranging from point mutation to chromosome breaks. Okay, the ionizing radiation such as gamma rays, once again repeat for you, the ionization radiation, particularly gamma rays mostly found it. The X-ray depends on the energy of the radiation, can create free radicals that result in the problem ranging from point mutation. Suppose it is create the free radicals, as a result what happened, the particular nucleotide will be, what is that, changed. So that condition is called as, what is the, you know, point mutations. Okay, to chromosome breaks. As a result, what happened, the chromosome will be break. Non-ionizing. So another one is called as opposite to the ionizing radiation, non-ionization. So such as ultraviolet radiation primarily affect the DNA. Already you know about ultraviolet is maximum. They split the component. So many chemical components. Take an examples. Ozone layer, O thrice is also split into O2 plus O. Okay, likewise, DNA molecules also split by this ultraviolet radiation, primarily affect the DNA by the creations of the thymine dimer, TT, which occurs when two thymines are adjacent on the strand of DNA. This is a non-ionization. Okay, so this is the another point. And UV radiation can cause the formation of a covalent bond between the two thymines, which prevent their participation in base pairing. So two thymines, what happened, you know, thymine always combined with the molecule of adenine, isn't it? Suppose thymine join with together, as a result, what happens now? Another participation, it will be completely prevented. So as a result, the mutations occurs in the chromosomal structure. This is the another one. And next, we will pass on to the next one is genes. Okay. So what is my genes? What are the types of gene is there? Okay. So the region of the DNA that transmit information to RNA are called as genes. DNA to RNA, the information, there is a mutual understanding between these two components. There is a language between this transmission of information to RNA are called genes. The type of DNA, what are the types of DNA is the multi-genes, repeated genes, single copy genes, and housekeeping genes, otherwise called as constitutive genes. These are the type of genes present in it. The type of genes, particularly multi-genes, means you know about the heading, there is an answer is there. Multi-genes. Multi means more than one. Repeated means same molecules, same codons repeated again and again and again. Single copy of gene means only one copy of gene present in the chromosomes. Housekeeping gene, always when there is a changes occurs, there is a permanent genes also present and also maintain. So that is called as constitutive genes. These are the types of genes is that we are going to see one by one, my dear. Okay. See that? The multi-genes. Okay. These are a group of similar or nearly similar genes for meeting the requirement of the time and the tissue-specific product. Example, globin gene family. Okay. Globin gene family is an example of multi-gene. This is the ancestor globin gene. So this one is the small blue color is the, this is the ancestral. Okay. This is the first category. The second category, what happened now, see the changes is that there is a light blue is in between. And the next one is alpha and beta. Alpha divided into two this side, alpha. Again, there is another symbol is here. The alpha again divided into psi 5, psi 2, psi alpha 2, psi alpha 1, psi alpha 2, like this. Likewise, these are all similar names, multi genes, same sequence, but different fractionation is there. Likewise, beta also, epsilon, lambda, and beta. Beta contain, see that psi beta, and delta, and beta. This one is called as GY and A lambda. Sorry, these are the different types. These are all are called as multi-genes. Each one it has a specific properties also. Okay, this is the another things. And repeated genes. What is the meaning of repeated? Repeated means again and again. It's multiply copy of genes. Okay, see that these genes occurs in multiple copies. For example, adenine, like that. Guanine. Okay, cytosine, whatever it may be, you can think about yourself. Repeated copy, multiple copy, 100 to 2000, up to 2000, in between, how many numbers is that? They are all a single copy of what is that? Nitrogen bases. But it repeated again means what happened? It is multiple copies. Example, they have given us a histone genes, tRNA genes, and rRNA genes, and actin genes. These are all are considered getting known as repeated genes. I have chosen here two examples. rRNA, so this is nothing but the ribosomal RNA. It has a two subunit. One is a large subunit, and another one is called a small subunit. It may be vary, depends upon the organism. Suppose if it is prokaryote, 70S ribosome is there. Suppose if it is eukaryote, what is it? 80 or 90? Media. Varsa?
Sir.
Yeah.
So is it 90, sir?
Yes, very good. So very good, exactly correct. That's why I ask you. Okay, so similar structures also here. The tRNA. This is the structure of tRNA. This is the structure of rRNA. These two are very essential for the translation process. When you are going to study the translation process, okay, so we can see the important points. Okay, good enough. Single copy of genes. Already we discussed. These genes are present in a single copies. They form 60 to 70% of the functional genes. Only one gene is that single gene only present. How many percentage? Entire organism containing 70% of the genes is single copy of genes, functional aspects. Okay, so this is the single copy genes. Next one is housekeeping genes, otherwise called as constitutive genes. Okay, housekeeping genes and constitutive genes. All genes are found in all cells. Some genes may express only in liver, some only in kidney, and still others only in the reproductive tissues. Okay. It contrasts to such genes, there are common set of example genes function that are, what is that? It forget here. I'm not able to see here. Okay, so such genes are known as housekeeping genes. Their functions refer to the housekeeping or constitutive activity because they are product or require for normal cellular activities. Okay, so these are all our, what is that? Constitutive gene, otherwise called as housekeeping genes. So wait a minute, I will show you. So what it has inside is that. Use so constitutive genes. Okay, just a minute, Ajay. I will come back. See that. Yes. I will come back. See, this is the type. See that housekeeping genes, constitutive genes, all genes are found in all cells. Some genes may express only in liver, some only in kidney, and still other only in reproductive tissues. Contrast to such genes, there are common set of expressed gene functions that are needed in the all cell types. This is called as housekeeping genes. See that such genes are known as housekeeping genes and their function is referred to as housekeeping or constitutive activity because they are products or required for normal cellular activities. Okay, this is the housekeeping genes. And non-constitutive genes, otherwise called as luxury genes, is also present. It is another part. Okay, these genes do not always express themselves in a cell. They are switched on or off according to the requirement, on or off, just like a switch. If you want fan, you can switch on it. Suppose if you no need, what happens? Automatically you will off. So likewise, some of the genes also act as a, which one switched on or off according to the requirement of the cellular activities. Such genes are of two types, inducible and repressible. Okay. Suppose that gene is present, what happens? It may induce some substances for the activities. Okay. And some of are repressible. Repressible, suppose if it is overproduction, automatically what happens? Some other repressor cells also activated. As a result, the hormones or any one of the component, it will be, the production will be stopped. Okay, see that first one is called as inducible genes. Okay, the inducible genes, see here, these genes are switched on in response to the presence of chemical substances or inducer. So that's why it is called as inducible genes. Suppose, take an example, see any one of the particular reactions has happened. Okay, during that reaction, what happened, when we are adding the chemical substances or any other inducer, just like an enzyme, what happened? The particular gene is active. Okay. Suppose we are adding over amount of inducer, over reaction happen. Suppose the reaction is very high. If you want to stop, means you are adding repressor. Okay, see that, which is required for the function of the product of the gene activity. Example, nitrate for nitrate reductase. So enzyme, the nitrate reductase is the enzyme. They require, which one in nitrate is a substrate, otherwise called as substance, otherwise called as inducer. Without nitrate substrate, there is no activity of the nitrate reductase, and as well as the lactose for lactopron concept, Jacobian monad. So this is also already we will discuss later, because already we saw it in the intro only, lactopron and tryptopron, like that it comes. So here inducible means that chemical substance induce the reaction. Okay. So the particular gene is also responsible. That is very, very important. There are two things as given. One is nitrate reductase, another one is called as lactose for the lactopron concept. Okay, see that one. So this is called as what is the lactopron concept. Okay, so here we will discuss there are three types of activities is there: lactose present, repressor inactive, opron on. We will discuss this point. Okay, here the promoter gene is there, structural gene is there, and also another component, inducer, promoter, inhibitor, that things is also. So here corresponding enzymes also secreted. Okay, the lac jet Y A, jet Y A, jet, it will synthesize beta galactosidase, permeases, transacetylases. Corresponding genes, they secrete the corresponding enzymes. Okay, this is very, very important one. Okay, so these things, just to note it down, we will discuss later very deeply. Okay, lactopron concept, gene expression, that chapter we will discuss deeply. Okay, don't forget, my dear. This is an example. Repressible genes, these genes continue to express themselves till chemicals, often an end product, inhibit or repress their activity. That's why I told you. Suppose if you're adding the inducer, the reaction will be continuously going on. There is no any end, infinity like that, that time what happened, automatically repressible genes also activated simultaneously. As a result, what happened? It will be stop the product. Okay, see that. Inhibition by an end product is known as the feedback repressors. Inhibition means what happened? Completely stop it. Stop the reactions. So that reaction is called as what is a feedback repressor, because due to the chemical substances, what happened? It may be inhibit the particular reaction. Example, tryptophan opron. So that one is called as lactopron. Here, tryptophan opron. This is the amino acid. Tryptophan is one of the amino acids, aromatic amino acid, phenylalanine, tryptophan. These are all aromatic amino acids. This was stopped. This is an examples of feedback repressor at the step. Which gene is responsible? Repressible gene is responsible. Okay, so this is very, very important. See the trap, TRP opron, so tryptophan opron. So the off and on. So these are the components mostly present. The tryptophan structure is here. So just like a switch off on off, opron status, this is opron status. On means what happened, off means what happened. Okay, so this is the another example. Suppose exceed what happened. See, the next one is a transposons genes or jumping genes. This is very, very important, my dear students. So many times they were asked the question, jumping genes. What is meant by jumping genes? The segment of the DNA that can jump or move from one place in the genome to another place. Take an example, A strand is there, and also B strand is also there, C strand is also there. The A strand, 49th position or 50th position component or genome transfer to the third one Okay, so the condition is called as what is transposons or jumping gene from the one place to another genome, one place to another genome. Okay, are called as jumping genes. Transpons were first discovered by Barbara McClintock for maize. Okay, this is very, very important. The jumping genes or transport genes was first discovered by the name of the scientist is called as Barbara McClintock from maize. Okay, this is the correct one, maize only. Another one is called as overlapping genes. What is the meaning of overlapping? One above the others. These genes overlap other genes, example, psi into 174. These are the two types of virus, psi into 174 viruses. These viruses, we can see overlapping genes. Overlap means over one over the another one. Okay, these are the another type. Next one is called as transcriptions. Already we discussed. What is by transcription? Transcription, nothing but the DNA to RNA. A particular DNA molecule synthesize RNA. With the help of the enzyme, what is that? RNA polymerase or DNA polymerase. What is that? So, RNA polymerases. Then only the RNA will be synthesized. See that transcription is the mechanism by which a template strand of DNA is utilized by specific RNA polymerases to generate one of 3 different classifications of RNA. This is very, very important. The RNA polymerase is essential for the synthesis of RNA molecule, and RNA, not only that RNA, the type of RNA, types of RNA. These are 3 RNA classes are as follows. Transcription, DNA to RNA, with the help of the enzyme RNA polymerases. How many RNA is present? You know, they exist in 3 forms. One is called as mRNA, also called as messenger RNAs. Second one is tRNA, that is transfer RNAs. Third one is rRNA, that is called as ribosomal RNA. These are the 3 RNA molecules present in it. Okay, mRNA, tRNA, rRNA. Okay, got it? Okay, see that. First one is called as mRNA, otherwise called as messenger RNA. What is the meaning of messenger? It has some specific codon, encoded the particular protein or any message from the DNA present in the nucleus. It has some requirement. The requirement will be passed on in the form of a message through RNA. So which one carry the message? That RNA is called as mRNA. Okay, it is transferred from or moved from the nucleus into cytoplasm, because translation process, the protein biosynthesis process takes place in the out of the nucleus, that is called a cytoplasm, because the cytoplasm contain amino acid pool. Amino acid pool is, what is the meaning of amino acid pool? There are 20 amino acids scattered numerously in the cytoplasm only. Okay, see that. These RNAs are the genetic coding templates used by the transitional machinery to determine the order of the amino acid incorporated in an elongation polypeptide in the process of translation. What is that meaning? You know, so it has a message. The type of protein molecules want to be produced. The message comes from where? You know, DNA. Okay. It is also one of the polypeptide chain, elongated polypeptide chain. It has a codon. Okay. And also mention the amino acid sequences also. Okay. So this is that one. So this is nucleus. Okay. Nucleus, it's contained the center, there is a DNA molecule. The DNA molecule produce mRNA. The process is called as transcriptions. With the help of the enzyme is called as, what is it? RNA polymerase. Okay, this is the process. It comes from where? You know, from the nucleus. It's enter into the cytoplasm. See that one? This one is called as cytoplasm. Okay. So here the mRNA and rRNA and tRNA is also there. The tRNA, it will come during the translation process, protein biosynthesis. The three RNA molecules is very, very essential. Then only the translation process occurs. Otherwise, it will not happen. Okay, so this is the process. mRNA, where it comes from nucleus. Okay, with the help of DNA to RNA, so mRNA, with the help of RNA polymerase. Because RNA synthesis takes place, that's why RNA synthesis, RNA polymerase. Okay, see that one. See that DNA to mRNA. So here the RNA polymerase is involved. As a result, you will get single standard only. See, they also has that genetic codes. See that one. And tRNA, another one is transfer RNA. These small RNAs form covalent attachment to individual amino acids and recognize the encoded sequences of the mRNA to allow correct insertion of the amino acids into the elongation polypeptide. It is very, very important. For example, messenger RNA is having some codon. The messenger codon is exactly attached with the tRNA carrying the amino acid sequences. These two is very, very essential for the elongation of the polypeptide chain. That is very, very important. Elongation. Elongation means leading of polypeptide, particularly protein molecules. Okay. So during this process, what happened? The correct insertion. Insertion means what is that? Adenine, thymine, guanine means here below there is a uracil and a adenine and also cytosine. Exact copy of the amino acids. Then only it can be paired. Then only the protein molecules will be formed. Otherwise, what happen? It will not happen. Okay, correct amino acid sequences is not possible. Means what happened? No, the required protein molecule they cannot able to synthesize. So the metabolism as well as other activities also completely what happened? Collapsed. Okay, we need a keratin protein. Okay, imagine keratin protein is essential for the hair and also nail also. Instead of keratin protein, they synthesize some other protein like as a heparin or globulin or myoglobulin like that, actin. Okay, what happened? What is the use of that one? Okay, that is very, very important. That was done by which one, mRNA and tRNA. This is the concept. See that one? So this is the structure of the tRNA. Already you studied. It has a 5 arms. The structure of tRNA, it has a 5 arms. One is called as acceptor arms. Here, the amino acids accepted by T loops. Right side. This is also the structure is also asked in the NEET point of view. Okay, which one it present T loop, and opposite there is a D loop. A, D, and T loop. This is also a small protrude-like structure is there in between the T loop and also anticodal loop. That is called as variable loop. It's a small extension is there. Okay, so how many arms is there? This is the acceptor arm, T arms, variable arms, anticodon arms, and also D loop. So okay, so the anticodon arms are connected with the messenger RNA with the triplet codon. These two setup completely inside the ribosomes. Okay, so the 3 ribosomes, RNA molecules, very, very essential. 3 RNA molecules is very, very essential. That is the things. The next one is rRNA. We will discuss that things is very deeply. RNA, ribosomal RNA. This RNA are assembled together with the numerous ribosomal protein molecules. Ribosomal RNA is also one of the RNA molecules. They are all assembled together with numerous ribosomal protein to form ribosomes. Okay, see, so this is a ribosome structure. 2 ribosome is there, 2 subunit is there. One is a large subunit. This is considered as a large subunit. This one is considered as a small subunit. They may be differ, depends upon their eukaryote or prokaryote. Eukaryote, it contains already about 70 S. Prokaryote, it contains 70 S. Eukaryote, it contains 80S. Okay, 2 subunit is there. So this is a long thread-like structure. It is called as messenger RNA, mRNA. Okay, this is the ribosomal RNA, a large subunit as well as a small subunit. In between, there is a small T-like structure. This is called as cloverleaf model. Okay, this is tRNA. So the 3 RNA molecules is very, very essential for the translation process. Okay, so this structure tRNA, there is a codon. Triplet codon is the anticodon is present in the messenger RNA. So particularly tell us the messenger RNA, it carries a triplet codon, and the anticodon is present in the tRNA. It collect the codon from the cytosol, otherwise called as cytoplasm. Okay, so this one is added, and when it comes, the bond formation takes place, it comes out free. Again, it entered to the next codon. So we see the next one. Again, it continued like that. Repeated and again and again. How many molecules, how many amounts, how many numbers they will need until the complete production of the protein molecules. After the completion, what happened? The stop codon will come out. Automatically, what happened? The protein biosynthesis will be stopped. So that is the, we will discuss one by one. Okay, this is the another point. See, tRNA. Okay, this is a L-shaped RNA is that. We will discuss cloverleaf model is also there. L-shaped tRNA molecules also present. There are 2 types of molecules. This one is called as mRNA. It has a genetic code. This one is called as ribosomal RNA. So mRNA, ribosomal RNA, and the tRNA. These are all are very, very important. Each one it has a codon. AA1, AA2, AA3 means what is that? It's nothing. Amino acid 1, amino acid 2, amino acid 3 like that. Different amino acids. Okay. The movement will be bad of the ribosomes. Okay. Ribosome, it can move in this way. Okay. The chain will move on opposite side. The tRNA also move on to the chain side. Okay. This is very, very important. And next one is during the synthesis, which one is very, very important? RNA polymerase is very, very important. Okay. Without RNA polymerase, what happened? No, the DNA molecule, they will not produce the RNA. Different type of RNAs. The transcription require an enzyme to catalyze the synthesis of the RNA molecules. The enzyme response for this is called as RNA polymerase. Synthesis of RNA molecules. First one is primase from DNA. Replication is an RNA polymerase. Primase is also one of the RNA polymerase enzyme. Okay. However, the enzyme for transcription is named RNA polymerase. Primase is also called as, what is that? RNA polymerases. All the enzymes. Here you must note it on one thing is very, very important, my dear students. The protein molecules are enzyme molecules. All the enzymes are protein. All the enzymes are protein molecules. Some of the enzymes may be hormone, protein, lipids, steroid hormone, like that. It's there. It is that. It is it. But here, the primase is a RNA polymerase. Think about that. Okay. So RNA polymerase, otherwise called as primases. Okay. The RNA polymerase require a DNA template. It is also require nucleotide triphosphate. The RNA polymerase, just like a DNA polymerase synthesis, only in the 5' to 3' directions. The RNA polymerase, just like a DNA polymerase synthesis. Okay. Only in the 5' to 3' directions. All RNA polymerases are depend on a DNA template, also called antisense strand, negative strand, non-coding strand to synthesize RNA. This is the difference. All RNA polymerases are depend on the DNA template. That means it synthesizes from the DNA only. Without DNA, there is no RNA molecules production takes place. Okay. Also called as antisense strand, negative strand, and non-coding strand. Antisense means opposite manner. Negative means, you know what about. Okay. Negative means, what is that? Acid. Okay. And non-coding means, what is that meaning? There is no, they will not indicate any amino acid. It's a particular nonsense codon. Already we discussed this in it. This is very, very important, my dears. Okay. So go to the next one. The resultant RNA is therefore complementary to the template strand of the DNA duplex, the identical to the non-template strands. Okay. So the resultant RNA is therefore complementary to the template strand of the DNA duplex. DNA and RNA always duplicated to each other. Okay. And identical to the non-template strand and identical to the non-template strand also. This is very, very important. And the non-template strand is called as coding strand, also called sense strand, positive strand, and coding strand. So this is very, very non-template strand is also called as coding strand. Coding means they mentioned. The coding strand is called as positive strand. Non-coding strand, negative. Okay. And sense means what is it? They mention some amino acid. There is a sense. The sense denotes amino acids. Okay. Or peptide or codon like that. Because its sequence are identical to those of the mRNA. However, in RNA, U is substituted for thymine. That's in uracil substituted by thymine. Okay, this is the important point of this part. Okay, this is a slightly complicated one. Just to you should understand and read it, we will get the ideas. Okay, see that one. So next one is transcription unit. Transcription unit means start site, where there is a starting place, where there is a terminator. In between, what are the things is there? See here, the promoter is the structural gene is the template strand is the terminator is also there. This is the coding strand. 3' to 5', 5' to 3'. This is the structure. Sometimes they may ask the question in diagrammatical questions. They may give the diagram without these parts. This is the first square is called as promoter. Okay. And second one is terminator genes. In between, the structural gene is also there, and also a template strand is also there. This is a coding strand. Okay. So this is collectively called as what is the schematic structure of the transcription unit. You see the next diagram, you can easily understand. See that one. 5' to 3', 3' to 5'. Starting there is a template strand, here non-template strand. So the first one is promoter. Suppose without promoter, what happened? The leading strand is not possible. Here the transcription site. Here the transcription site is the started from here and the end with this part, that orange color. Okay. So this site is called as terminal. In between this part only the RNA coding region. So this is called as transcription unit. This area only. Called as what is a transcription unit. Okay. So you will get RNA molecules. I think this part is finished, my dear students. Okay. So I will summarize all those things once again for you. See that one. So you will finish it up to this level because it is very complicated and also very inconvenient in nature. So, okay. So mutation already we discussed. Chemically induced mutations. What are the chemical substances they induce the mutation? Who discovered first? The chemical mutation was first noted by Charlotte Auerbach. Okay, where mutagenic effect of the mustard gas when they were used in the chemical weapon in the World War I. So what are the chemical substances means? First one is called as nitrous oxide. The nitrous acid is deaminating agent. Deaminating amino acid removal of the amino group from the particular component. Cysteine to uracil, guanine, xanthine, adenine, hypoxanthine. Okay, so this is the point. So is the conversation. See that amino group is replaced by the oxygen. The condition is called as deamination. Deamination is nothing but the removal of amino group. So as a result, the structure also changed, and also the chemical names also changed. Cytosine, see, converted to uracil. Okay. See the cytosine and uracil. And sugar and here also sugar. See that methyl cytosine, here thymine. Change. Okay. Here the amino group is replaced. Here oxygen is there. Here is also amino group replaced. So you will get it this one. This one cytosine. This is methyl cytosine. Converted to uracil and thymine, respectively. Okay. So alkaline agent already but methyl group is there, and also ethyl group is also replaced. As a result, what happened? You will get it the addition. Sorry, so donate alkyl groups. Alkyl groups we are giving to the component. So as a result, what happened? The ethyl methyl dilphonate, dimethyl sulfonate. Dimethyl nitrosamines. These are the formation. Addition of these two components. What component? This is the newly formed component. Bases analogs. Bases analogs already know about is changes already know about the base analogs chemicals with structure similar to that of the any one of the 4 standard bases: adenine, guanine, cytosine, thymines. What are the structure they are having? Similar chemicals or bases. Another one substance also that they can easily replace. Okay. The DNA polymerases cannot distinguish these analogs from the standard bases. They are having only standard bases. We can easily identify. Okay. So this is another point. So 5-bromouracil already know about the 5th carbon atom. The bromo is also utilized. So as a result, the changes of the things. Normally 5-bromouracil pair with the adenine just as a thymine does, but it occasionally mispair with the guanine, leading to the transition. Instead of thymine, they may be guanine. So this is the mismatch or mispairs. You see that one structures. And second one is called as 2-aminopurine. So transition is, this is the normal process. It can be replaced by mispair like this. Instead of thymine, replaced by the cytosine. Okay. This is the another one. Acridine dyes. It's a heteroaromatic. Okay, what are the heteroaromatic flat molecules present in it? Acridine, acriflavins, fluoroflavins, eflavins, acridine orange, ethidium bromide. No, don't forget, my dear student. This is very, very important for the DNA fingerprinting. Ethidium bromide, the color will be in orange in color. We can easily expose to the X-rays, you will get orange color. And also dioxin. Okay. How it will be means addition and deletion. Already you know, but there are two things: the addition and deletion, and of the mutation is called as frame shift mutation, otherwise called as gyprasic mutations. This is very, very important. So this is a structure, already we saw it. And hydroxylamine, what is the meaning hydroxylamine? And it's also addition with the hydroxylamine and cysteine, hydroxylamine cysteine is formed. So these things also already we discussed. Radiation, ionization radiation, non-ionization radiation. Ionization means X-rays comes out. Non-ionization means it depends upon their thymine dimers only. Thymine dimers means 2 molecules of thymine molecule present in it. As a result, it may be different. Okay, ultraviolet radiation is also there. And genes, the types of genes already know: multi-genes, repeated genes, single copy genes, housekeeping gene is also there. What is meant by multi-genes? More than one genes, alpha and beta, particularly an example is globulin gene family. Okay. So another one, you know, repeated gene means tRNA, mRNA, sorry, tRNA and rRNA genes and active histone. This is very, very important, my dear students. Repeated genes, given examples means you don't forget this one. Next one is single copy of gene. Most of the cases, 70% of the gene is single copy. Housekeeping mean and constitutive genes is also already known, but it's also mostly present in the organ systems, liver, kidney, and also reproductive tissues. Okay. So the such genes are known as housekeeping genes. What is the meaning of that one? You know, keeping or constitutive activity, because their products are required for normal cellular activity. It's very, very essential. So it's commonly present as it is without any changes. Okay, just like a TNIIT. Okay, it's present. Non-constitutive genes, luxury gene is also there. So it is divided into 2 types: inducible and repressible. The inducible means when they need, that time it may be activated. When they do not need, that time it will be off. So just like a switch off. Okay, that is inducer. Inducer means that, take an example, some nitrate is an inducer for the nitrate reductase. Lactose is an inducer for the lactoferrin concept. Okay, so that's why it is considered getting known as inducible genes. The particular gene induce the reaction and catalyze the particular enzymes. So that is a things already we saw it, the things also. And repressible genes means it is stop the activities. Okay, inhibits the activity after the complete productions with the presence of the chemical substances. Example, tryptophan opera. Tryptophan operon is there. Okay. And next one is transport genes and also jumping genes. Jumping gene is already you know about it's jumping from the gene from one polypeptide to, and then third one or fourth one, whatever it may be shift on to the 10th place to 25th place, like that. This was first discovered by the name of the scientist Barbara McClintock from mice. Overlapping genes, give an example. This is the examples. Don't forget, my dear students, psi into 174 viruses. Psi virus as well as the 174 viruses is also very, very important. Don't forget. And transcription, there are 3 mRNA is very, very essential. Okay, mRNA, tRNA, and rRNA. Okay. So we'll finish it up to this level, my dear student. Remaining things we will see on the next classes. Thank you, my dear students. Thank you, Ajay. Take care. Thank you, ma'am.
sir.
Thank you, ma'am.
Thank you, sir.
Thank you, ma'am.